The first topic is by booking Weinstain. Hi, Brooke. Could you?
Hi. Hello, sorry, I changed my computer configuration to show my Signathed print in the back, and so I'm all kerfuffled. Hi, it's Brooke, and thank you for having me.
Okay, let me introduce Brooke Weinstain. She obtained her bachelor's degree in biology from Georgetown University, and her master's degree is in cellular and molecular biology from San Francisco State University. She is currently a PhD student at the University of California in Merced. Her research interests include using comparative genomics to study the evolution of introns and genomic complexity across eukaryotes. And the fun facts about her is she'll ask. Okay. Welcome, Brookie.
Great. Were you going to go ahead and play my video?
Yeah, you can play your video. Let me play for you. Sorry.
Great. Thank you.
Sorry.
Oh, no worries. Thank you.
I was sharing. Okay. So let's begin.
Everyone, my name is Brooke Weinstain, and I'm a PhD student at the University of California, Merced. I'm very excited to share the results of my analysis on differential alternative splicing in Gulf pipefish brains during male pregnancy. For my PhD, I normally study intron evolution, but obviously, like everyone here, I really love Signathed. So this is a nice opportunity to combine two of my favorite things. And I'm excited to share the results with you today. Male pregnancy is perhaps the most extreme form of male parental care among vertebrates, as we know, particularly in Signathus pipefish, where there's a very well developed extensive brood structure. Males provide nutrients, protection, aeration, and osmoregulation to the developing embryos. And we also know that the brain regulates many processes associated with pregnancy, which I think makes it particularly exciting to look at gene expression in Gulf pipefish brains in response to male pregnancy. I have gene expression here instead of alternative splicing, like in my title, because I also did do a gene expression analysis as well, So the alternative splicing is looking at kind of a qualitative approach to the transcripts, and then we have a general regular quantitative approach with the gene expression analysis as well. So this is a quick overview of alternative splicing. It's a critical regulatory mechanism in eukaryotes that produces diverse, often tissue-specific and often functionally distinct proteins to maintain normal cellular functions. It's also widely implicated in many diseases and disorders, including cancer. So there's a lot of increased attention on it. This is what constitutive or regular splicing looks like, where we have 4 exons that are joined together by introns, shown as these lines here. During the splicing process, these introns are removed from the pre-mRNA transcript. The exons are stitched together to form the mature mRNA. In alternative splicing, we basically have more than one mRNA transcript resulting from the same pre-RNA gene, from the same gene. So we get more than one protein potentially from the same gene. And there's a handful of different major types of alternative splicing, ways that we classify them. I'm going to run through them very quickly. We have exon skipping, where we either have the inclusion of a cassette exon, illustrated here in green, or not. You can see the top mRNA transcript shown here does not have that cassette exon, whereas the bottom isoform does. We have something called an alternative 5' splice site, which is what it sounds like, where we see on the top usage of a splice site downstream of the canonical splice junction, and this results in a second exon that is shorter than the canonical exon. We also have the opposite, where we have an alternative 3' site that's used. We have something called intron retention, where an intron is retained in the mature mRNA transcript. This often results in protein degradation, but also can result in a second functional protein. And then we have mutually exclusive exons, where one or the other of an exon is included. So I'm just going to go through my methods really quickly. I used the annotation file and the genome file from the Gulf pipefish genome paper. I used RNA-seq reads that were 100 base pair paired end reads. I did not generate these reads. I just did the bioinformatics on this project. So I want to take a minute to thank, during the talk, people who generated this data, which were Dr. Emily Rose, who's at Valdosta State University, and Dr. Adam Jones, who's at the University of Idaho. They were generous enough to let me use it for their analysis. I also used a pipeline that's a little bit different from traditional methods of looking at alternative splicing, because I wanted to optimize detection of novel splice junctions. I used STAR, which is a very splice-sensitive read alignment, and a special technique called two-pass alignment, which is outlined here, which basically separates genome indexing and splice junction discovery from the quantification stage. So once I had the reads quantified, I did further quantification and actually defined junctions with JUM, which is the junction usage model. It's a new algorithm that's also was created to optimize novel junction detection. I also used JUM to actually do the differential splicing analysis, including multiple testing correction or using an adjusted p-value. And then finally, I did a differential gene expression analysis using DE-seq2, which is pretty traditional. And I also use multiple testing correction here as well. So before I give the specific results of the differential expression between pregnant and non-pregnant males, I just want to give a snapshot of what I detected in regards to alternative splicing events globally in the brain tissue. So all the events that I detected were 1,765. They're divided 4 different types of alternative splicing, comprise the bulk of these. The alternative 5' and 3' sites are the most heavily represented. We have a lot of intron retention and a good chunk of cassette exons. This is a little bit unexpected because in most vertebrates, we see significantly more cassette exon usage than other types of events. So this could be an artifact of my analysis, or which I don't think, or the data, which is possible, but also could be something interesting that's going on in cygnathids. So cygnathids do have tend to have reduced genomes compared to other vertebrates. They have shorter exons. Maybe they've shifted to doing something differently with their alternative splicing. So this would be something that I'd like to look into in the future. So I'll show the GO gene ontology analysis. These are the top molecular functions for all of the alternative splicing events that I detected. You can see we have a lot of binding activity. There's probably a lot of transcription factors in here. We have a lot of enzyme activity, particularly kinases. These are proteins that modify other proteins. We have a lot of calcium ion binding, which we know is important in pregnancy. Almost 30, I guess 35 calcium binding genes. We also see almost 20 calmodulin binding genes. This is a calcium receptor. It's just a nice, I think, overview that's interesting of what's going on before we dig into what is differentially spliced or associated with male pregnancies. This is that same figure for reference. And then this is what I actually found was differentially spliced. So I found 21 events, and the breakdown is a little bit different. I think most notably, we don't see a lot of intron retention anymore. We do see the most cassette exons, which is a little bit more in line with what we would expect, but also still almost as many alternative 5' sites. So I'm just going to show you what a few of these look like. So I think it's cool to see. These are sashimi plots that actually show the mapping of the reads to these different splice junctions. This is the Drebin protein. This is, I'll talk about this as a pool gene in a second, but what I'm showing you down here, so this is the intron exon structure from the annotation, and in this case, it matches where the exons are actually mapping. And then these lines that we see are the median number of reads in each sample that are spanning this particular junction. This is separated by non-pregnant and pregnant males. This is like a snapshot of the junction in the average non-pregnant male, and like a snapshot this junction in the average pregnant male. And what you might have noticed is that while pregnant and non-pregnant males exclude this cassette exon at about the same rate, we do see about a 13% in the inclusion level of this cassette exon in pregnant males. The strebin protein is interesting. It's an action cytoskeleton organizing protein. It's known to play a role in the formation of cell projections. It's important in the regulation of memory-related synaptic plasticity. Pretty interesting protein. The last cassette exon I'm going to show you is the cold shock domain containing protein, E1. Here we see the same thing, a 13% increase in the inclusion level of the cassette exon in pregnant males. This is exciting to me, especially because it's a multifunctional RNA binding protein, and it's known to orchestrate cellular proliferation and differentiation, kind of fitting with seeing the remodeling of brain tissue in male pregnancy. So this is the first of two alternative splice sites that I'm going to show you. This is in protein MAX. You might see that on the bottom, I'll start with that, we have a read that matches up with the canonical exon junction down here. And on the top, in both non-pregnant and pregnant males, we see an alternative 3' junction that's upstream of the canonical junction site. And this is there's also a 30% increase in the usage of this alternative site in pregnant males, so 31 reads on average compared to 12. Protein MAX is interesting. It's a well-known transcriptional regulator. It also antagonizes a very famous oncogene, MYC, which is implicated in cell proliferation, differentiation, and apoptosis. And then the last gene that I'm going to show you before moving on to the gene expression analysis is a Fragile X mental retardation protein. I'm most excited about this for two main reasons. One of which is that we see usage of an alternative 3' site only in pregnant males. So you can see here, only in the pregnant males, we see this upstream splice site being used. This is going to result in a shorter second exon than usage of the canonical splice site. This is also very exciting because it's a very widely studied gene. FMR1 in humans is essential for normative cognitive development and female reproductive function, which is interesting when you're thinking about male pregnancy. It's also one of these, so because it's essential for normal cognitive development, it's also very implicated in a lot of diseases and disorders, probably most famously autism. There's been a tremendous amount of research done into this because of that and other disorders that it's been linked to. So this is a paper that just came out this year. In zebrafish, there have actually been quite a few studies in fish that have knocked this gene down and have found mostly behavioral changes. Like in this study, they found changes associated with anxiety, hyperactivity, and stereotypy, which is habituation in zebrafish that have had the FMR1 gene knocked down. So this is very exciting, and I'm looking forward to looking into it further. So I'll jump into the differential gene expression analysis results. I separated them by genes that are upregulated during pregnancy and genes which are downregulated during male pregnancy, which I'll show you on the next slide. So I found 9 genes that were significantly upregulated. I think they're all pretty interesting, but I'm only going to point out a couple of them. The first is CYP450 or chrome P450. These are enzymes that are involved in the metabolism of both endogenous and exogenous compounds. That includes drugs, environmental toxins, dietary things that they eat. Brain CYPs in particular are also thought to participate in the metabolism of some neurotransmitters, endogenous steroids, and neurosteroids, which I think is exciting. And then I'll also highlight this U2SURP gene, because proteins that are known to contain this SURP motif are all known to be involved in splicing. So I feel like that's kind of an interesting gene to be upregulated in a study looking at alternative splicing. Genes that are downregulated, not as clean separation between the samples, but these are still all sick. And I'm only going to talk about one of them, the one that I'm most excited about, which is aromatase. Which is a gene that I think we've all heard about in association with male pregnancy. It's necessary for the synthesis of estrogen from testosterone. There's a study that's been done fairly recently in monkey brains that shows high levels of aromatase in regions of the brain involved with reproduction. So that's pretty exciting, and another one that I'm excited to dig into further. Other studies in fishes with male brooding have shown decreased levels of androgens like testosterone during the parental phase. So this is kind of in line with that as well. So I'll quickly go through the gene ontology, molecular functions for these two different groups of genes. So we have on the left the differentially expressed genes, and on the right we have the alternatively spliced genes. And we can see that these are very different functions. Pretty much they're all different. We have our aromatase here. And one that I'm interested down here is we have insulin-like growth factor binding, which I think is kind of cool. But I'll just leave it that we have very different groups of functions being regulated here. So in conclusion, what I just said, differentially spliced and differentially expressed genes between brain tissue and pregnant and non-pregnant males are independent, except for one gene, which I will share in a minute. Before I do that, I do want to say this initially to me seemed really exciting, and I still think it's exciting, but it has been shown in other studies, including other studies in fishes, like this one in salmon, which found, basically the same thing, that gene expression and alternative splicing play contrasting roles. So it's showing this kind of orthogonal or perpendicular suites of regulatory mechanisms. So that one gene is NAT16 so an ectotherm. There's not that much that's been shown about it in fishes, but there are some recent studies. One of it shows an ectotherm homologue of it that's predicted to be responsible for NAH synthesis. This study also found 2 splice variants in fish. I think it's mackerel and a couple of other fish species. Those expressed exclusively in brain and eyes, which was nice to see. And they also proposed that the function of mammalian and NAT16 has been altered from NAH synthesis to another different biological role. They are not sure what that is. In conclusion, we see, I think, a good general thing that we can feel good about saying is that we do see functional overrepresentations consistent with nervous system remodeling via differentially expressed isoforms and alternative splicing in response to male pregnancy, which I think is pretty cool. And before I do my acknowledgements, I'll also say future work. I think the next exciting thing to do is to do comparative work, both within cygnathids and also within other vertebrates, particularly other more well-studied fish species, and also to look into what these isoforms are actually doing, if they're coding for functional proteins, kind of what the downstream effects are a little bit more concretely. So with that, I will conclude and thank my advisor, Dr. Scott Roy, who supports me doing this work, even though it is not directly related to my PhD thesis. And again, I want to officially thank Dr. Emily Rose and Dr. Adam Jones for being kind enough to share this data set with me to work on. Thank you to the organizers for organizing this conference and putting it together. It was a highlight during a really difficult time. Thank you, and I'm happy to take questions either now or via email.
Okay, thank you. Thank you, Brooke. Does anyone have questions? Okay, I have a small question about the gene of N86. Do you have any idea what details of this gene involved in the last gene you have showed?
Sorry, which one was it?
N86. Yeah. I think you have found you have selected it out, right?
Yeah, off the top of my head, I don't have anything to say about that, but I do have all of the Go analysis results very handy. So I'd be happy to touch base with you and let you know what that gene has turned up as. Are you asking about kind of the functional associations with that gene? Yeah, I'm sorry, I don't know them off the top of my head.
Okay, thank you so much.